Wow, look at all these jellyfish.
Eh, they look pretty boring. They aren't even doing anything.
I guess you're right.
Wait!
What?
Before you go judging jellyfish, you should know about how useful they really are.
Yeah, they're actually incredibly important to one of the most important scientific discoveries.
You should watch this video to learn more about the green fluorescent protein and how they come from jellyfish. GFP, which stands for green fluorescent protein, is a fluorescent protein that has found many uses in research due to its ability to fluoresce when exposed to ultraviolet light and its stability in many mammalian cells. This protein first existed around 116 million years ago in a species of jellyfish known as Achoria victoria, where it gives off a green glow. This form of the protein is sometimes known as AVGFP. AVGFP has an excitation peak at a wavelength of 395 nanometers, with an emission peak at 509 nanometers. It is also 238 amino acids long and has a molecular mass of 27 kilodaltons. Early studies of AVGFP showed that the protein was able to fold and fluoresce at room temperature and did not need exogenous cofactors specific to A. victoria. However, it also has several drawbacks as the protein has dual peaks of excitation spectra, is sensitive to pH and chloride, and has a poor photostability and folding at 37 degrees Celsius. Further research has also shown that GFP is resistant to detergents, proteases, and drastic temperature changes. The first big mutation that enabled the use of GFP in biological research was first reported on by Roger Chen in 1995. This is a single point mutation, S65T. This resulted in improved spectral characteristics such as increased fluorescence, photostability, shift of major excitation peak to 488 nanometers, and a peak emission at 509 nanometers. Subsequently, enhanced GFP was discovered in 1995 by Thastrup and Falco labs and is a point mutation, F64L, that resulted in improved efficiency at 37 degrees Celsius. This allowed for the practical use of GFP in mammalian cells. There are also mutant colors that have been discovered, such as blue, cyan, and yellow. GFP has a beta barrel structure with 11 beta sheets and a pleated sheet arrangement. There is also an internal alpha helix with a covalently bonded chromophore. The beta barrel is 42 angstrom long and 24 angstrom in diameter. Internal side chains of the beta barrel induce a cyclization reaction that induces chromophore formation. The H-bond network and electron stacking interactions of the internal side chains influence the color and photostability of GFP and its derivatives. GFP has found many uses in the scientific field and outside science in other fields. In the biological research field, GFP has been primarily used as a reporter assay. An example of such a use as a reporter assay is being a reporter for the environmental toxicity of various chemicals. GFP has also been used in fluorescent microscopy and provides the advantage of not being phototoxic when used in live cells. Some other uses of GFP include its use in microscope photography and use in transgenic animals or pets, which results in the pets fluorescing under UV light.
The tool GFP is the reason why we have those stunning images of glowing brains and cancerous tumors nowadays. But how does this protein, something we usually think of as being invisible, actually glows? It comes down to the structure that looks like a barrier and a chemical reaction that acts like a tiny light switch. Let's start with the structure. GFP is a protein found naturally in species of jellyfish. It is a long chain of 147 amino acids. But in biology, shape is everything. This long chain doesn't stay straight. It folds up into a very specific shape. Imagine you took a length of string and carefully coiled it into a perfect cylinder. That's what this does. It creates what we call a beta barrel. It looks like a soup can made of protein. The outside of the can protects the inside, and inside of this can, we can have a secret ingredient. Right in the very center, 3 specific amino acids, a serine, a tyrosine, and a glycine, are held tightly together. They are locked in this dark, protected chamber, waited for a trigger. The trigger is oxygen and a little bit of energy. When these 3 amino acids are forced together in the presence of oxygen, they undergo a chemical reaction. They bond together to form a new structure. We call this new structure chromophore. This is the engine. And here's the mechanism. You shine a light on the cell, specifically a blue or ultraviolet light. The chromophore absorbs that blue light. It acts like a sponge. When it absorbs that energy, the electron inside it jumps to a higher energy state. But physics hates an unstable electron. They immediately want to snap back down to where they started. And when they snap back down, they release that energy. But they don't release it as heat, and they don't release it as blue light. Because of the exact structure of that chromophore, they release it as a lower energy, which is a green light. The barrel around it is vital here. It stops the energy from vibrating away and forces it to come out as a visible light. This mechanism is brilliant because it is self-contained. The protein builds its own light bulb. In the 1990s, scientists realized they could steal the gene of this light bulb and insert it into other organisms. They attach it to a cancer cell, and suddenly we can watch the cancer cell move in real time.
Hello, my name is Gurjinder Singh, and I will be covering the history of GFP. So GFP stands for green fluorescent protein. It is actually one of the most useful tools in modern biology. It was first discovered in the early 1960s in a glowing jellyfish called Aquaria victoria. Scientists were basically trying to figure out why this jellyfish glows, and during that process, Osamu Shimomura isolated the protein responsible for the green fluorescence while studying the jellyfish's light system. What makes the GFP really cool is that it doesn't need any extra chemicals to glow. The protein literally makes its own light-producing center called the chromophore from 3 of its own amino acids, serine, tyrosine, and glycine. This happens automatically inside the protein's beta barrel structure, which protects the chromophore and helps it fluoresce reliably. The big turning point came in the 1990s when Martin Shalfie showed that the GFP gene could be inserted into completely different organisms like the E. coli and the C. elegans, and the cells would still glow green. That was huge because it proved GFP works on its own and isn't dependent on the jellyfish. After that, Roger Cyan engineered improved versions that were brighter and even different colors, which made GFP way more powerful for imaging. Once scientists cloned the GFP gene, it basically became a biological highlighter. Researchers could attach GFP to proteins and actually watch where those proteins move inside living cells in real time. Because this completely changed biological research, Shimomura, Shalfie, and Cyan won the Nobel Peace Prize in chemistry in 2008, and to this day GFP is still used everywhere in molecular biology and biochemistry labs.
GFP creates huge contributions to human society. Before GFP, studying processes inside living cells was extremely difficult. Scientists often had to kill cells, stain them, and observe static images. It could see structure but not the dynamic processes in real time. However, GFP had changed that. Because GFP is encoded by a gene, scientists can attach the GFP gene to another gene of interest. When that gene is expressed inside a living cell, it produces a glowing protein. This allows researchers to watch proteins move inside living cells in real time. For example, scientists can track where a protein travels, whether it goes to the nucleus, the membrane, or other places. They can observe how cells divide, how neurons grow, and even how cancer cells spread. One of the biggest impact of GFP is that it made cell biology dynamic. Instead of taking snapshots, we can now record movies of life happening at the molecular level. GFP also led to the development of many fluorescent proteins, including red, yellow, and blue variants. This means scientists can label multiple proteins at once and observe how they interact. Also, GFP has lots of contributions in healthcare. For example, it is used in brain mapping, where scientists use GFP to label specific neurons, allowing them to map brain circuits and track neurodevelopment. In addition, it is also used in cancer surgery for tumor imaging. Scientists engineer tumor cells to express GFP in animal models, so when surgeons operate, the cancer tissue glows. which helped them to see tumors' boundary clearly. Overall, GFP is a game changer that transformed modern biology and medicine. What began as a glowing protein in a jellyfish has become one of the most powerful tools in biotechnology, proving that even a small discovery in nature can create a massive impact on human society.
So, what did you all think of the video?
Wow, I didn't know that GFP and jellyfish were so important.
Yeah, learning about the mechanism behind the fluorescent was so fun.
And this is why you should always tell people about important scientific discoveries.